Thank you for joining us this evening for the spring installment of our Forensic Science Lecture Series. The series was established way back in 2004 to help educate the public in the forensic sciences and related fields, as well as to spotlight the fascinating stories that scientists have to tell. The lecture series is sponsored jointly by the Indiana Medical History Museum and also what is now, but wasn't at the time, the University of Indianapolis Human Identification Center. Is that still what it's called?
Yes.
Okay. I feel like there for a while, every time I looked, I was wrong. So I'm glad. Delighted to introduce you to our speaker, Dr. Amandine Eriksen, who is making her second appearance here on the IMHM stage. If you've been coming to events here for quite a long time, you might remember we did the math 14 years ago, back in 2010. You weren't a doctor then. Amandine was here with Dr. Stephen Naraki of the University of Indianapolis, and they did a program on her graduate work analyzing gorilla skeletons, sort of non-human forensic anthropology. You might mention that.
I will probably.
So I'll stop talking about that, but I did want to mention that she's been here before, and it's fabulous to have her back. Dr. Eriksen is an assistant professor in the biology department at the University of Indianapolis with expertise in anatomy and osteology. Her research has taken her around the world studying skeletal asymmetry in humans and gorillas to better understand how stress affects the primate skeleton. Please help me welcome Dr. Amandine Eriksen.
Thank you so much. All right, well, that was a wonderful intro. So today I'm going to focus a little bit on what a lot of my recent research has focused on, in which I'm very interested in stress, right? And how stress actually affects and changes our skeleton. Let's see if this actually does work.
No?
All right, it might not work. Oh, it's okay. I'll just hit the button. Okay. So obviously Sarah gave a great intro, so I don't really need to introduce myself. The one thing I will point out that I think is wonderful in this image, obviously some of us experienced a total eclipse on Monday, which was amazing. And I took the liberty of taking our University of Indianapolis logo, UIndy, and making a little dotted sign so that the lunar, the solar eclipse could shine through. And so what you're seeing are little half suns right there on that sign. So I just thought that was a fun thing that I had to show everyone. All right, so let's get to this. What is stress, right? When we think about stress, we often think about things that, affect both our body physically, but also our mind. And so we often think of stress as anxiety that we experience, sort of everyday type of stresses. We might think of illnesses that we sometimes experience as things that would, make us stressed out. But then there are also other things that can cause stress in our body, like not getting enough proper nutrition, especially for long periods of time, going through periods of having a lack of vitamins in our diet. All of these things essentially can collectively fit under this category that I call environmental stress. So it's how our environment is impacting us. And then we also see that there are other forms of stress, like genetic stress. And often this is going to relate to times where you just have maybe groups of people or really any animals where you might have a lack of diversity in the genetic material across that population. And so that in itself can cause stress at a genetic level as well. Now, the thing is, when we think about stress, we often think it's all bad, right? Well, I'm here to tell you there is some, I'm going to call it good stress. And what we see is that as humans, especially, we tend to do things like perform repetitive tasks, right? We use our right hand or our left hand usually as our dominant hand. And so what we see is there are other stressors that will kind of impact our body, right? We put pressure on our body when we use our right hand maybe more dominantly to do everyday tasks. And when you do that for essentially your entire lifetime from a young age, you actually do change the shape and size of your bones, where you have muscles that are attaching. And so, of course, there are other people that will preferentially maybe use their upper limb or maybe their legs, like dancers and athletes, who are also putting that biomechanical stress or load on those body parts. So I like to think of this as it's an interesting form of stress. It's not necessarily the bad types of stress that we often think about. And so when does this happen, right? When does stress actually impact us the most? I mean, I've been pretty stressful this week getting ready for this talk and everything. Did that really change my skeleton? Probably not. But what we see is that especially during early life, that's when stress is going to have the biggest impact on our skeleton when it's growing. Okay, and so what we see is that in early life, even before we're born, when our bones first are developing prenatally, that's when we're going to see that stress potentially being able to impact the skeleton the most. It doesn't stop there. We also do, continue to grow and develop after we're born, so postnatally. And so we see that there are childhood stresses that can also impact the bones as well. So what do I look for in the body, right? When I'm looking for signs of stress, I'm really interested in two specific things. So my specialty is in skeletal asymmetry research. So I'm really interested essentially in how different the right side of the body is from the left side. So in the image at the top of the slide, you can see that we have two skulls essentially. So we're looking at two faces. And you'll notice that there is a dotted line that essentially separates the right from the left side of the face. And so in this image, even if you really don't know that much about bones, you can look at the lower jaw and you can see that it's, both sides are not just a mirror copy of each other. You notice that the jaw is kind of shifting a little bit. It's a little bit wider on the left side, or sorry, on the right side of the image for both of these individuals. And so we're seeing that there's some asymmetry, right? The right side is not exactly the same as the left side. And then we also see that stress can appear in the form of linear enamel hypoplasia. And we see this specifically in teeth. So not only can your bones affect or be affected by stress, your teeth can be as well. And so in the image above, you can see that there are these kind of horizontal, they're slightly curved, but there's these horizontal grooves in the teeth. And those are essentially growth defects. So what happened is while these teeth were growing, this is an adult, so we're looking at adult teeth. While these teeth were growing, this individual went through some sort of event that was stressful enough that it caused growth in the teeth to stop, just for a little bit. But it was enough that once it started back up, you are left with this impression essentially that shows that growth arrest. And so with this individual, you can see there's actually more than just one groove. Each tooth has at least two, and they are very worn down. But you can see a second groove on most of the teeth. So that indicates that this person probably went through more than just one stressful life event throughout their childhood, when their adult teeth were still growing and developing inside of their jaws. So I couldn't give this talk and not tell you about a little bit about gorillas and why I really got interested in all of this. When I was younger, I was able to get involved with a project in Rwanda where we studied mountain gorilla skeletons. They're obviously different than humans, except their bones look fairly similar. So being a human osteologist expert, it was really easy, honestly, to transfer over to studying gorillas for a little bit. And I really got interested in especially asymmetry research when I met this gorilla. So her name is Taina, and Taina here, you may notice that her face looks a little contorted, and she's not actually making a face. This is what her face looked like in real life, and you can see that her nose is not perfectly straight. And so you may assume that Taina maybe had a troubled life, maybe she would have had trouble finding a mate, or maybe she would have had trouble reproducing, but she did not. She's actually the youngest gorilla in the wild ever to give birth, which is pretty amazing. And she's actually holding her infant. You can see her infant's head right here that she's sort of hugging. Now, unfortunately, Taina did pass away at a fairly young age. She did have a congenital heart defect, and so that ended up being the cause of her death. But the one, it's like kind of interesting just thing about her is that because she did pass away early, we were able to look and see what did her skeleton actually look like. And so this is a 3D model of her skull. And so you can see maybe in this image when you're looking at her from the front, or this one here where you're looking down at the top of her head, you can really appreciate how shifted her, especially her mouth is, right? Her lower face is definitely kind of contorted in a way that it's not, it's definitely the bone itself that's impacted. And you can see this in the image of her when she was alive. So she's definitely the whole reason, honestly, why I really got interested in, like, why is this happening, right? And so what we see is that it is really important to study stress, because we see that there are a lot of species out there that are at risk of extinction. And it is really important for us to find ways to mitigate that. And so being able to study stress, look at how stress, especially, gorillas and other animals are, it does give us a good idea as to why we should work really hard to help with conservation efforts in protecting these animals. The other thing is that understanding stress, especially in living primates, so ourselves, even gorillas, chimpanzees, our closest cousins, it also gives us a lot of insight into how people before us survived and lived their lives. How do they deal with, with certain stressors that we still potentially deal with? And so it does make it a little bit easier for us to kind of reconstruct how our past, human ancestors would have lived their lives. Okay, so I want to talk a little bit about what is asymmetry? How do you find it? How do you see it? What does it mean to actually be asymmetrical? Okay, so this is your task right now. I've got two images up here. And what I want you to do is look at both of these and try to figure out which one of these looks like it's more asymmetric, which one of these looks like it's more symmetrical. And while you do this, I will tell you, one of them is incredibly asymmetric and the other one's actually pretty symmetrical. So there are differences. Now, the one thing I want you to do is try not to pay attention to the teeth down here, just because those were just broken after the person died. And so that's not part of that. We're not worried about that. But looking at their faces, I'll give you a moment to think about it. You can kind of pretend, like draw like a pretend line down the middle and try to imagine, does the right side look like the left side? You don't have to answer, but it'll give you a moment to look. Okay, it's not easy to tell, right? I mean, I study this and even I struggle to see it's difficult. So I'm going to make it a little bit easier. What I'm going to do is I'm going to essentially take the image of one side of the face, and I'm going to essentially take that image, I'm going to flip it over onto the other side. And what we're going to see is, you notice, I'll just use this pointer here, you notice this kind of faint purple line along both sides. And now you can really appreciate that on the left side, we have, that's the one that's going to be a little bit more asymmetric, right? And you can actually see how much wider one side of the face is on this individual compared to the other side. It's a little bit more narrow versus the skull on the right. You can see a much fainter purple line. And so that's implying that that individual is a lot more symmetric. Did you get it right? Maybe? It's okay, you can all say yes, I believe you. Okay, so here's where I'm gonna get into a little bit of nitty gritty, giving you a background on what types of asymmetry exist. So I'm gonna keep this fairly basic. I won't lie, some of it's a little complicated, but I'm gonna do my best. And so the two main types of asymmetry that I'm interested in study are fluctuating asymmetry and directional asymmetry. And we can kind of parse these out in that fluctuating asymmetry is going to refer to these small random changes where you may see that either the right side of the body is a little bit bigger or the left side. It's random. It could be either side. And usually there's small fluctuations. That's why we call it fluctuating asymmetry. And often we'll see that this type of asymmetry sometimes is linked to, there's a lot of previous research that's shown that this is linked to, I'll say like a good health, right? Often people have shown that when you have individuals that have a low amount of fluctuating asymmetry in that population, in that group of humans, sometimes it's been thought of as a signal that that population is in better health because they have those low amounts of fluctuating asymmetry. We'll get back to that in a moment. And then we have directional asymmetry. So from that, you can kind of tell things are going in one direction or potentially another. But what we see is that consistently we see one part of the body being larger than the other across the entire population. That's sort of the key here. You have to look at what is happening across a group of people or gorillas, if you will. But you have to look across that group to see, is this something that you're seeing in everyone? Or are you just seeing it, in one person? That tells us a lot about what's happening. Now, there's a part of, well, there's many parts of our body where we actually see directional asymmetry in ourselves. Think about your internal organs, right? Of course, the skeleton that we'll be talking about, we see that tends to be very symmetrical, but we see a lot of directional asymmetry in our internal organs. So think about, your lungs, for example. Yes, you have a pair of them, but you'll see that one side you have 3 lobes, the other side you have 2, right? They're not exactly the same. And then think about your liver. Your liver is located predominantly on the right side of your body. So you are programmed to grow asymmetrical, right? It doesn't necessarily mean that there's something wrong with the individual when there's the presence of these asymmetries. And so we'll talk a little bit more about each of these in humans. So how do we actually identify asymmetry? Well, obviously, I showed you pictures of skulls and you tried to figure out, how asymmetric they were. It was incredibly difficult. But what we can do is essentially compare the left sides and right sides of each single bone or paired bones. So obviously you all spend a little bit of time looking at the cranium and sort of thinking about, how do you see the right and left sides of that when it's a bone that we have kind of in the middle of our skeleton. But then here we have an image where we have paired bones. So these are femurs that are thigh bones. And in this image here, they may look very similar, but I tweaked them a little bit so they're a little different. But if we were to compare these bones, we would just compare the right femur with the left femur and essentially try to calculate how asymmetrical, how different that right femur is from the left. Now, the one issue that I run into is that when it comes to asymmetry research, we don't all agree on things. And what we see is that there are some conflicting sources when it comes to trying to determine what does asymmetry mean? Where does it come from? What causes it? And so what we see is that because of that, a lot of researchers, when they have these different ideas as to what these types of asymmetry mean and where they arise from, it ends up leading to a lot of conflicting and different interpretations, which causes some problems because I might think something means something and another researcher might interpret it in a completely different way. And so when I first got really interested in asymmetry research, I started reading a lot of articles and trying to, solve all of life's puzzles about asymmetry, which there's a lot, there's enough that I could write a whole dissertation on it. And one of the things I found was there was this hypothesis that's called the good genes hypothesis. And it's sort of controversial because essentially it, indicates that, especially fluctuating asymmetry, the presence of it is sometimes by some previous researchers is sometimes linked to having these kind of this higher quality of DNA, having better genes essentially. And we see that some anthropologists that study this will often think of these good genes as indicating that individual is kind of in better health, or that maybe they were able to essentially buffer themselves from whatever stressor they were experiencing, even at the prenatal, the fetal period. And so maybe that means that individual is fitter. And so what a lot of people did to try to prove this was they did these attractiveness studies. And what we see is that a lot of people, a lot of researchers that started studying attractiveness, right, would essentially assume that because usually in nature, ideally symmetry is perfect, right? And so we would assume that something that is the most symmetrical is the most perfect, which means it has the best genes and it's the most beautiful. And so I found this picture of George Clooney. I think everyone knows him, hopefully. And I mean, he's a very attractive person. No one can really deny that. And so what we see in this image, so the center image is showing their original photo of George Clooney. And then on either side, you essentially have his symmetrical image where they've taken either the right side and flipped it over to the left or vice versa. And I think when you look at all these images, it's hard to pick and say, the most symmetrical one is definitely the most attractive, right? It's hard to say that. And so what ends up failing with these attractiveness studies is we essentially don't find that symmetry is always the most attractive to individuals. And so that means that people that are even a little bit asymmetric continue to find mates, continue to reproduce. Thank goodness. So when you go home today, check yourself out in the mirror. You'll start to notice you have some little asymmetries as well. The other problem that we see with the current research into asymmetry is that the way in which people have been studying this, that has been very inconsistent. One of the ways in which this often happens is that researchers will often just pick one part of the body, right? You'll pick the face. Look at how attractive it is. Look at how symmetrical or asymmetrical it is. Or, for example, we could pick the sacrum. So that's the bottom part of our spinal column. It's the part that kind of is part of our pelvis. And so what we see is that there are a lot of studies that will pick one bone and they'll just try to figure out how, why is that asymmetric? What is happening? And this is such a great example. The sacrum, I won't pick on the researchers, but this bone has been studied by multiple researchers where they make these pretty grandiose claims where they measure this bone, they figure out, okay, it's asymmetric, and then they claim that they can predict hand preference. They can tell whether someone's right-handed or left-handed from measuring this bone. Is it potentially possible? Absolutely. However, the problem with these studies is that they're often conducted on individuals where we have no idea whether they're actually right or left-handed. We're just assuming because when we look across the world's populations, we do tend to see that there's an overwhelming amount of people that are right-handed. However, I don't believe it, or I don't buy it. Like, if you're going to study this, you have to study people where you actually know whether they're right or left-handed. The problem is there aren't a lot of skeletal collections out there where we definitively know whether individuals are right or left-handed. But don't worry, I found one. And so in this case here, with the sacrum, we see that these researchers, again, were saying, if the sacrum was larger on one side, they could predict that that person was going to be right or left-handed. So in comes sort of where my research stems from. So one of the things that I wanted to do is I wanted to look across the skeleton, right? Instead of just picking one bone and studying that one thing, I thought, well, how about I pick like multiple bones? When I do the whole skeleton, I can't. There's 206 bones. It's a lot. But I picked the largest bones that I could think of that I thought were, especially bones that other people had studied in previous research. And I thought, okay, I'll look across those bones and I'll see, do I see the same pattern of asymmetries across the bones? So for example, if this person has a really asymmetrical head, do they also have an asymmetrical pelvis? Do they also have a really asymmetrical, thigh bone? Is it all, if we see evidence of directional asymmetry, is it all going in the same direction? Okay. And then I thought, okay, well, let's say there are different patterns, right, where I don't see the same bones having the same types of asymmetries. Why is that happening, right? And so I came up with kind of two big things that I would sort of test. And so I thought, okay, if I see big differences in asymmetries across the body, I want to figure out, could it be that hand preference, right? That biomechanical stress that we're putting on our bodies, could it be that that's impacting that stress that I'm seeing, especially if it's directional asymmetry? Or could it be that because our bones, we think about our bones growing, yes, some of our bones will start appearing and turning into bone in our little fetal skeleton around the same time, but they go through very different patterns of when they start to get longer. Some bones will finish growing a lot sooner than other ones, especially when we get into childhood. And so I thought, well, maybe I could look at how certain bones, if they share a very similar time period during which they're developing, maybe those bones, maybe they'll share the same asymmetry patterns. Maybe that could explain things, right? But you never thought you'd come here and just talk a lot about asymmetry, but don't worry, we'll get to some linear enamel hypoplasia a little bit later. So to carry out this research, I went to 4 different universities or museums where they had skeletal collections that range, where we had some individuals that were more modern, that had died as early as the 2000s, or as late as the 2000s. And then I had some much older populations that had died around the 1900s. And so overall, I sampled 123 skeletons. One of the challenges is that I didn't want individuals that had lost their teeth, only because when you do end up losing your teeth or having them extracted before you die, that ends up kind of changing the mouth just enough that I thought that might end up changing my results in terms of what asymmetry I saw. And so I thought, I'm going to stick with individuals where I don't have to wonder if some of the asymmetry I'm seeing is just because they've lost some of their teeth. The one neat thing is that there is a skeletal collection in Tennessee where individuals have been donating their bodies for years now. And those individuals, when they donate themselves before they pass, they end up actually filling out a form in which they provide their own self-documented hand preference. They exist, I found them. And so I was able to go to this collection and sample some of these individuals. Of course, 40, I wish it were more. But when you start looking for people that have skeletons where maybe they haven't broken bones, I mean, who hasn't broken a bone? Definitely not me. And so that did kind of it did make me narrow down my sample, but I was still very happy to find these individuals. So you can see here all the different bones that essentially I looked at across the skeleton. So I'm going to take you through a little bit of my data collection process. I may skip through a few slides a little bit because I know we might not have as much time today, and it takes me a while to talk to you guys about asymmetry, but I'll give you an idea of what I do. So I use a method in which I study all of this with 3D bone models. So the first thing I have to do is I have to go get that 3D bone model. I have to create it. And so I spent a lot of time just sitting in basements of museums and essentially 3D scanning bones with my structured light scanner. And from that, I was able to make these wonderful high resolution 3D bone models. What's amazing about this is that you're then able to, well, I was able to do some asymmetry research, but you're able to reuse these individuals in terms of trying to analyze different things. And what you can see is one of the methods in trying to estimate asymmetry that I had to use was essentially placing these landmarks. So those little black dots that you see with all little numbers there, those are landmarks. And I think you can see my highest one this is actually not a lot of landmarks, 49. So in total, I would put 49 of these little points on this 3D specimen within a software. And from that, I essentially was able to be able to estimate essentially the size of those bones and figure out whether or not the right or left side of different parts, in this case of the face, would be different. So what's neat here is you have what's called like a little wireframe. I would create these just to make sure that I didn't have a crazy amount of outliers, because sometimes I may put a landmark in the wrong place. That's my fault. And I don't want a computer program to say, oh no, this person's really asymmetric, when it means that Amandine just put the landmark in the wrong place. So I would use this, and so I just think it's a fun visual. And so what I'm going to do is I'm going to overlap all 123 specimens for you in 1 second. And there they all are. So it's kind of neat. You're seeing the array of every single person that I had in this study all kind of overlap. So I think it's just a fun visual to be able to see how much variation there is between people. Okay, so this is where I'm going to skip a little bit, but I promise you, if you want me to give you another, I can spend a little bit of time later on talking about how we actually measure fluctuating asymmetry, but I'm going to get to my results because that's the fun stuff. Okay, don't worry about this. We'll just keep going. There's a lot. Okay, there you go. So we're going to get to my results. So when I looked at fluctuating asymmetry, one of the things I was able to do is essentially create these percentages that would tell us how much of that deviation from right or left, how much of that was due to fluctuating asymmetry itself. And so from these percentages, what I found was that in the arm, we saw that there were very high levels of fluctuating asymmetry in the arm bones. And I found that there were very low levels of fluctuating asymmetry in the parts of the skull and that sacrum in the pelvis. So what does this really mean? Well, first off, it shows that things are different, right? I wasn't seeing the same pattern of asymmetry across the skeleton. I was seeing it was high in some places, low in others. And so overall, I didn't see any significant correlations between all those bones. That if I saw a really asymmetric skull, I didn't have a really asymmetric pelvis, okay? So that means now I had to go and look at those alternative kind of reasons to try to explain why do we see these differing patterns. So one of the things I wanted to do was, and I'm going to skip a little bit over the growth pattern stuff, but this is what I thought would be kind of interesting, especially to you folks, because we're in the Medical History Museum. And what I see with the individuals that I collected, because a lot of them came from these museum collections where they often had been sort of donated through anatomical programs, those individuals often were people that were kind of the unclaimed dead. They weren't necessarily people that had families to claim them. And so one of the things that we often see is that within these skeletal collections, a lot of these individuals usually are people that would have had a low socioeconomic status in their life and potentially at the time of their death. It also, signals that they might have been people that had kind of developed under those low socioeconomic statuses earlier on when they were potentially experiencing stress. And so one of the things that I thought would be really interesting is looking to see, do I see kind of these historic or temporal changes across the specimens that I had? Because with those 123, we see that there are some people that lived and died in the early 1900s, others in the 2000s. And so my prediction was that when I looked across these individuals, right, if fluctuating asymmetry really is a sign that people are experiencing or high levels or shows that people are experiencing a lot of stress, I expected that people that had lived and died a long time ago would have really high levels of fluctuating asymmetry. And so what I did is I grouped everyone into these three categories from kind of oldest to more recent. And interestingly, I did find that the fluctuating asymmetry levels were quite high in the humerus, so that upper arm bone in the oldest group. However, I found that the percent of asymmetry across all the groups, was actually the highest in the most recent groups. So yeah. So what does that mean? It could mean a lot of things. I think it means that we're still experiencing stressors, right, in different ways. So maybe our stresses are not dealing with malnourishment in the 1900s, but we might be dealing with, we're still dealing with things in our lives, even in modern day. So now I'm going to move on to directional asymmetry. So here's the really easy way that you measure this, right? You take a measurement on the right side of in this case, the pelvis, take a measurement on the left side, subtract one from the other, plug everything into this formula, and you get a number, right? You get a score, essentially. You have directional asymmetry, and then you can take it across the bones. You can average those scores, and you get kind of one composite directional asymmetry score for that bone, and then you can compare those across the skeleton. Because I was doing 3D stuff, it got a little bit more complicated. There's my landmarks, there's the amount of measurements that I technically took. It's a lot. We won't go through all of that. But in the end, I ended up still with that composite directional asymmetry score. So what do we learn? Well, when it comes to directional asymmetry, I saw two places where I saw this positive correlation. So in the arm, if the right humerus was bigger on the right side, I saw that the right radius for that individual was larger on the right side as well. When I looked at the skull, I saw that the same pattern was happening where when the skull was asymmetric on one side, right, I saw that it was also asymmetric on the other side. And so overall, when I looked across all of the skeletons, this is everyone, all 123, I did find that on average, we did see that these, the arm and the skull were both consistently deviating towards the should be pointing this way, towards the right side of the body, that they were larger on the right side. What does this relate to? Well, obviously with the arm, that could relate to hand preference, because we do see that individuals are often more right-handed. With the skull, what about chewing side preference? Have you ever thought about that? When you put food in your mouth, especially if you're right-handed, sometimes you tend to actually put it between your teeth on your right side, right? So chewing preference is actually a side preference, is a thing that does exist. Now, was I right? I wasn't positive. So what I did is I went back and I looked at my 40 individuals where I knew whether they were right or left-handed, and I was right. So what we see is that here we have a really nice example. This is a person who's left-handed. So this is their arm bone on both sides, on the right and left side. And I put a little box around it so you can really appreciate that you really, even with the naked eye, right? You didn't have to measure this like I did. You can even see that difference. Look at how much shorter that right side, that right humerus is. So overall, I saw that there were high levels of that directional asymmetry in the arm and the cranium, like I mentioned earlier. I saw that there were also high levels in parts of the pelvis too. And so the main point of this is that, yes, biomechanical stress is definitely something that's impacting our skeletons. Could be hand preference, impacting the arms, could be chewing, impacting our faces. And so those do strongly influence directional asymmetry. So in the end, I looked across those bones, found that yes, absolutely, both asymmetries definitely differed across all of the bones. And then we were able to tell that yes, in part, some of it does relate to hand preference, some of it does relate to the timing of bone growth. I know I kind of skipped over the bone growth, but I promise you, I did the research, it's showing that. And I can show you later another slide if you really want to see. So how am I doing on time? 6:17. Okay. I'm going to skip, well, I'll skip the linear enamel hyperplasia. What's really neat is even you two can measure enamel hyperplasia, look at the screen, and just try to count out how many grooves you see. I'll even help you. So looking at the incisor, we're just going to count those grooves. So looking at that one incisor, we at least see two really clear ones, right? I've angled the light so you can see the grooves a little bit better. On the tooth next to it, I see at least 4, right? This is a very rudimentary method where you just take a picture and you count. There are other methods that are much more complex where you can essentially cross-section the tooth and look at it under the microscope, and that's going to give you a much more accurate prediction or actually accurate measurement. Bless you. And so, of course, my prediction was that people that died a longer time ago would have experienced more stressors during early life, so they would experience more linear enamel hypoplasias. And that is exactly what I found in that oldest historic group that I created, people that had died within, between 1912 and 1939. Those individuals did have the highest amount of linear enamel hypoplasia, almost twice as much in terms of how severe they are. So in this picture here, you can see someone without any of those grooves. And at the bottom, you have someone with a very severe, very deep bruise, which implies a more stressful event happening. So the one caveat I will say is that I didn't have a huge sample of these individuals with severe linear enamel hypoplasias from that old group. So I think some of that could potentially be altering, biasing things a little, but it is nice to see things, come out the way that you did predict. Okay, I'm going to skip over talking about genetic stress, even though I love this, but I'm going to skip over talking about gorillas. Enjoy the lovely images of gorillas. They live in Central Africa. This is the project that I was a part of. And overall, spoiler, we see high levels of fluctuating asymmetry in mountain gorillas compared to other gorilla groups. And that's because they have experienced so much, they've been endangered for such a long time. They've experienced these huge decreases in numbers, and essentially that's affected them in that they don't have a lot of genetic diversity, okay? And so that's causing that genetic stress to occur within that group. And when we compare mountain gorillas to other gorillas, we see that other gorillas do not have very high levels of fluctuating asymmetry comparatively. And the thing is, they all live in pretty similar environments. They all have a similar, access to really high quality food throughout the year. And so essentially we can kind of see that the environmental stressors are pretty much the same across the groups, but it's those genetic stresses that are different. And so that's one time in which we can actually show what's happening. It's harder to do in humans because we move around and we don't always live in these tiny groups. Okay, so in the end, yes, asymmetry definitely varies across the skeleton. Biomechanical stresses are definitely impacting asymmetry. And then with the linear enamel hypoplasia, it's not necessarily the same events, stressful events that are causing those as what is necessarily causing fluctuating asymmetry, because we still see high levels of fluctuating asymmetry in modern humans as well. So in the end, if you take anything away from this, I think there are a lot of applications to understanding more about stress in general. And we see that obviously it has roots in being able to help give us some evidence as to why we should continue conservation effort for endangered species. When it comes to human history, there obviously are ways in which we can study asymmetry and those linear enamel hypoplasias to learn a lot more about our distant ancestors and recent ones as well, and learn a little bit more about, what did an early human experience? Did they have, did they live through stressful events that were as stressful as what we're dealing with today, right? And then finally, there are some forensic applications as well, because sometimes it would be nice to at least add to, if you've got an unknown skeleton, it would be nice to add and say, well, this person was likely right-handed. Or really, it would probably be more beneficial to say this person is likely left-handed, because that might be a more unique sign that might be able to help identify that person. All right, so I'm gonna stop there. Thank you. I'm gonna end on a sunset because I once had a student tell me that's what you should always do, and it's pretty good advice. So
thank you so much. We have a little bit of time for questions. I have just one from the online audience, so I'll go ahead and throw that at you.
Okay.
Jane Schultz asks, is it possible to see differentiation when traumatic historical events occur, such as World War II?
Oh, that's a great question. Now, one of the, everyone heard that, right? I think. So one of the issues with everything I just told you about is that it can be really difficult to actually pinpoint what is that stressful event that's impacting someone. We can assume, that it might have been environmental stressors, genetic, but it is unfortunately, it is very difficult to narrow down that stress. One thing that we can do, and it's not necessarily exactly what I do, but we can sometimes pinpoint very specific life events. And our teeth are a really big key in that. So if we take our teeth and we essentially cross-section them, so we cut them in half and look at those at high magnification, you'll actually see your teeth have kind of these growth lines in them that are very similar to a tree, right? A tree has those rings, you cut a tree in half, you can sort of count back and figure out how old is that tree. You can do things very similarly with teeth. You can cut a tooth in half, and you can examine it and sort of count back. And you are often able to estimate when that specific stress event would have happened. Now, I love teeth, but I don't love teeth that much. So I don't do that research. I have colleagues that do that and they tell me all about it. But yes, it would absolutely be possible to kind of count back and figure out, did that person experience a really stressful event like that at that, around that time? Yeah. Mhm. Yes.
Got here late, so I may miss this. Racial differences.
Well, I can't attest to racial differences. One of the things that I did just essentially for ease of my study is I did not let that, that wasn't a factor for me, in that I only looked at individuals that were of white descent. I just did that honestly so that I didn't have to also add that in as a factor. But that is something that I'm definitely interested in exploring, and that would help increase my sample size too, and then that would be another factor that I would have to look at. It's a great question. Yeah.
I know you mentioned that just due to like some of the lower population sizes, you're starting to see like some higher frequencies of genetic abnormalities. Do you see it? Is there like an increase in things like congenital heart defects that you described tonight, or like other things that are causing issues because of low population size, or is the
Okay. That's a good question. So in case anyone didn't hear that, so when I look at across those gorilla groups, we have this kind of these small population sizes versus the big ones. Do we see a difference in those maybe those smaller groups? Are they experiencing higher genetic defects like the congenital heart disorder? I am not positive about that, mostly because when it comes to studying wild gorillas, you're at the mercy of whoever wants to live there and study the wild gorillas. And so luckily there are some wonderful people that go out and work with these conservation groups and will live overseas and monitor the health of gorillas. We just don't see this across every single group. So mountain gorillas are very well studied, specifically because conservation efforts have been so strong, because they are at risk. But then we have other groups that live in other groups of, especially in the West, of gorillas, where they live in these huge groups, and so they're not necessarily monitored as much, where we would have that kind of data necessarily. So that would be amazing if we could get that, but I'm not positive.
Could I ask them from that line?
Yes.
Sarah asks, what type of event causes linear enamel hyperplasia?
There are lots of things that can cause linear enamel hyperplasia. So it could be, now it does have to be something, an event that is stressful enough that it causes some of your bones and your teeth, when we're looking at these hypoplasia, to occur. And so that means that it has to be something where your body is going to essentially stop allocating resources to growing teeth and just make sure your brain and your spinal cord and all of that stays alive. And so often these will be events as severe as maybe having like a really, really bad bout of flu, or, you know, if you've got a mother that's pregnant and is experiencing maybe just unable to get good high quality sources of food for a really long period, even that potentially could impact. The crazy thing is that birth itself is a very stressful event for all parties involved. And so the birth itself will often actually leave. They might not be very, very visible to the naked eye, but it itself will actually often leave little tiny linear enamel hypoplasia on the baby teeth. So because those are forming, right, that enamel for your baby teeth are forming when you're born. And so that event itself can cause just a little bit of a growth disruption, which is wild. So check out your kid's baby teeth, or if you've saved any. Okay.
Chemotherapy is one of those stressor
Yes.
that they see on teeth, or starvation.
Mm-hmm.
And Holocaust survivors, they have those stressors.
Yeah, that's a perfect example. As long as it was happening while that individual was growing and developing their teeth, yeah, you would absolutely see record of that. Do you have follow-up?
Just using living people.
Oh, living. Well, I was thinking CT scans of living people. So I'm going to go and get those CT scans, because often we see that people like athletes tend to injure themselves, so they tend to have to get CT scans. The hard part is really getting access to those records, because then, you know, I've lucked out so far that I've had to deal with people that are deceased. It's not really lucky, but it's that I don't have to get, IRB permission to do all of that. So it does, there's an ease to that.
Excellent point, sure.
It does make life easier, at least for me. So I think I should have put everything, I did put the museum collections in my acknowledgement. So I do thank everyone that was part of my study as well.
Whenever you were talking about dividing your entire data set into subsets based on, I think it was years, how did you go about separating this? Was there a particular event that?
That's a great question. I'm trying to think if I really had a strong rationale. It's probably somewhere tucked away in my dissertation. For the people that lived, or at least the people that died within the 2000s, that was really easy because I essentially have this big gap between 1966 and about 2000. I don't have anyone because at that time, basically anatomical or like anatomy, gross anatomy courses stopped donating their cadavers to museums. And then we saw that we didn't really have body donation programs just yet where individuals were actually donating themselves to universities to become potentially part of their, their skeletal collection. So there's a big gap there. So that one makes a lot of sense. But yeah, I did have to find a way to sort of parse out people that lived, or I should say died between 1912 and 1966. And I think I just picked kind of like 1945 because, it was a nice, it's an odd number, but it made it so that I didn't have too few people in one category. So I can't remember off the top of my head, but I feel like I had enough people in the 1930s that I didn't want to have a really small group in the early 1900s and then a really big one in the middle. And so I kind of parsed them out just to make things a little bit more even across the group. So I felt like I was doing a more thorough investigation.
Any questions?
I've got more.
I just said, how did you do research on the samples that you, the samples that you were able to do? How did you do that research, or how did you kind of parse out if they had what you were looking for? Because you talk about a lot of exclusionary factors.
Oh yeah, I sort of gave myself these parameters where I didn't want anyone that was too young because I wanted adults. So I looked for individuals that were somewhere between the age of kind of 30 and under 70, mostly again because I thought that might exclude individuals that had a lot of osteoarthritis, right? Because that'll impact the bones a little bit, in which that might impact the measurements I'm getting. And so I wanted to make sure that the bones themselves were within that range. And then I got to the museum and I essentially looked through everything that they told me fit that parameter. And I just looked at every single head, every single body. And I looked for anyone, like I said, that had at least had their teeth when they passed. And then I looked for individuals that didn't have massive bone fractures and things like that, because obviously that would impact asymmetry. And I didn't want that to be something where I then wondered, well, is it because, this person broke their forearm and that's why the bone is 5, millimeter or centimeters shorter. So those were some of the things that I did. We have time for another.
Sure.
Do you still? Okay.
Are you interested in do you only use American institutions for your research?
Yeah, for the human research, obviously, I only did use.
Are you going to try to expand that research out internationally, or do you have colleagues who are studying the same thing internationally?
It would be nice to go and study this with international populations. Right now, I still have a lot that I could do in the US. So I think the next steps would be to continue doing work here, especially now that I'm in an institution where I have graduate students where I can maybe get them to help with some of those things. It would be because scanning bones does take a lot of time. And so I would love to, though, to be able to give this very expensive piece of equipment to a 25-year-old and make sure that they don't drop it. It's actually one of the reasons why I didn't buy a handheld scanner. I bought one that goes on a table so that it can't be dropped as easily. But I would love to continue sampling individuals that I have here just for the ease of travel and not having to have that expense. But I do have some colleagues overseas that have started scanning bones, but it tends to be one bone. Everyone loves the face, so it tends to be that the skull is what gets scanned a lot. So it would be still a nice comparison, but not everyone scans the rest of the skeleton. That's why I did this, because there just isn't a lot of research looking across multiple bones in individuals. We have time for.
Let's do one more.
I'm gonna pick Justin.
Oh, two. If
Oh.
you get two, let's not fight.
All right, in your experience, like any sexual dimorphism between the bones?
So yes, so the question is, was there a sexual dimorphism between the bones? So that means did we see a sex difference? And we are going to see that there is a size difference. So one of the things about this research is it also tries to focus on the shape differences as well. So I didn't go through the nitty gritty of exactly how I calculate those asymmetry scores, but part of it does take a little bit of the size factor out, so you can compare males and females and not worry that the size of the bones themselves is what's going to impact the score. Good question. Okay.
Thank you very much. And thank all of you for coming.